the complex cellular membrane. So this is actually what the membrane of a cell looks like when it's made really, really simple in cartoon form. So researchers have identified thousands, actually 40,000 different lipids now have been identified in cell membranes, not just one. Every protocell experiment, they use one type of lipid. Actually, a cell is made up of thousands of different types. Mixtures of monoacyl lipids destabilize the system. So when they have their monoacyl lipids, those will destabilize the systems. You can't use those. Lipid bilayers surround subcellular organelles such as nuclei and mitochondria, which are themselves microsystem assemblies. Each of these has their own lipid composition. So this is a lipid bilayer. You have some pointing out toward water, some pointing in toward the water on the interior of the cell. And the outside ones are different than the inside ones. Nobody knows how that was done. Nobody knows. Clueless. Nobody knows how that was done. Every protocell experiment just uses homogeneity throughout the whole thing. And so it's not really a protocell. It's not really reminiscent of a cell. Now, within each one of those, you have other organelles like nuclei and mitochondria that have their own bilayer assemblies with their own constitution, different than what's on the outside of the cell. Nobody knows how that was done. Plus, these have these proteins. So there's this non-symmetric distribution. Then there's proteins that go through here. These are ionophores. That allows certain things into the cell and certain things out. Without that, you can't let anything into the cell. It's going to kill the cell. It has very discrete sensors that allow certain molecules to get in, certain other molecules to get out. It has ionophores that allow certain ions in, certain ions out to keep the ionic concentration at a certain level for maintenance of that cell. As soon as that ionic concentration gets off, you know what happens? Boom! The cell explodes because the ionic concentration has gotten off. It blebs and explodes. Nobody knows how that is done. None of the protocell experiments, none of them have these proteins going through them as these control gateways.
Fact 2. Prebiotic forces cannot create the basic building blocks needed for the development of the basic cell. There is solid scientific evidence that you are designed. Come visit us at ageofdesign.org.